Well, this is my second attempt because we had somebody come to the door and interrupt the recording. But
Yeah.
Dr. C is here with me, and so he may chime in from time to time if you guys don't mind.
Yeah.
When we last left off, and this may be right after, if your brain didn't need a huge break, we talked about the visual system and its components, meaning the parts of the eye, from the cornea to the pupil and iris and the lens that projects onto the retina, where we found light waves are converted into a neural signal by the cells in the retina, specifically the rods and cones. Rods were more numerous, cones very few in comparison. We then went on to I went on to talk about the visual system itself and how information from light hits the back of the retina first when it bounces off the lens, and the rods give off rhodopsin, the cones give off iodopsin. This causes these chemicals to be transduced into a neural signal and allows for that signal to go from the bipolar cells to the ganglion cells, which we will get to in a second. The ganglion cell axons form the optic nerve, which leaves out the blind spot of the retina, heads towards the brain, crossing over about 85 to 90% at the optic chiasm, or making an X, and majority of the axons from the left eye go to the right LGN, lateral geniculate nucleus of the thalamus, and from the right eye, majority of those go to the left LGN, and from the LGN of the thalamus to the visual cortex, or V1, of the occipital lobe. We also talked about color vision. Color vision is mediated by the cone system with the smaller receptive fields. And they're much like the auditory system, where we had the tonotopic map, and we had two theories to review, which were place and frequency theories. We had two theories of color vision, one that centered around the cones themselves, the Young-Helmholtz trichromatic theory, which says that there are three types of cones, R, G, and B. But one thing that this theory has trouble telling us is the color yellow. Since the primary colors are R, G, and B, red, green, and blue, from those types of cones, it's really kind of impossible to get the color yellow just from that theory alone. So that theory, although it was a good one, was insufficient. So Herring, a few years later, came up with this theory called the opponent process theory, where it's not the cones, but it's how the cones specifically send their information to the receptive fields of the ganglion cells. And remember, the ganglion cells, as I just said, are the ones that send the information to the brain. So they are done in an opponent process fashion, where red and green are the opposites, and that's why you would have red-green color blindness. Yellow and blue are opposites. That's why you would have the second most common color blindness is yellow-blue. And black and white are opposites, although that one's more from the rods than the cones, which we're going to get into now. So the best way to think of both, to think of all of color vision, is like it was in chapter 9, like it was with the auditory system. That's why I talk about all these similarities. It is the combination of trichromatic and opponent process theories.
Question.
Dr. C has a question.
How common is color deficiency of red and green?
Red and green, I don't know the answer overall to how common it is in the general population. I would probably venture to guess about 10% of people, but red-green is the most common color blindness of the two types. Red-green is the most common, followed by yellow-blue. But this also goes to talk about that dress that broke the internet. Remember, the dress was seen as either black and blue or white and gold. And now we know that this is because of the opponent process, that you perceived it individually either as black and blue or as its opponent process, which is white and yellow or gold. Getting into what we're going to talk about this time around is form vision, which is more about input from the rods than it is from the cones, which deal with color vision. So when information comes from the visual system to the primary visual cortex, or what we know as V1, it's a very elementary type of information before it goes to secondary and higher order associations. In the last chapter, in chapter 9, we did talk about how A1, auditory cortex, was for sort of the more simple elementary sounds, and that for us to be able to understand multiple sounds and to understand different pitches and different instruments in, let's say, a band's performance, that we would have to combine it with higher order association areas of the auditory cortex. So A1 gave way to secondary auditory cortex, or A2. In this way, we have V1, which takes the primary input from the visual system, and then we have many other areas of association: V2, V3, V4, V5. V4 is actually what's processing the different colors inside the brain. So before I get to all of that, I'm going to back up and let's talk about form vision. So just like a tonotopic map was formed for the auditory system, we have what we call a retinotopic map for the visual system. So again, what this means is where this hits in the retina, and we did have that slide in the last lecture that looked at points A, B, and C and where in the retina it hits, closer versus farther away. What we see is where it hits in the retina, it's going to hit in an adjacent spot in the visual cortex, much like in the auditory system. So adjacent retinal receptors activate adjacent cells in the visual cortex. But this doesn't really necessarily explain form vision itself. What we really need to throw into the mix is contrast enhancement and what we call edge detection, where we can see the contrast of where one thing ends and another thing begins. This could be based on its color, it could be based on its shape, it could be based on how light or dark the boundaries are. And that's where we get into this idea that I started talking about in the last lecture a little bit, and we're going to get more into it here in this lecture. And to solidify it, I am also going to make sure that I have the video for lateral inhibition from YouTube also available as a link. So, when we talk about the ganglion cells are receiving information not just from the cones for color, but also from the rods for black and white and edge detection and things like that. And I talked about the ganglion cell fields in terms of color last time, but we can talk about them in terms of black and white for edge detection. So for the ganglion cells, we have what are called on-center, off-surround cells, and we also have the opposite of that, off-center, on-surround. What you see at the bottom of this slide is a black area, but in the middle there's a white, and it's got a little plus sign, which means that in the center of the cell, light is going to increase the firing, which means more action potentials and more information to the brain. In the outer side, in the lateral or surround area, it's all black, which means that it decreases or stops firing. So this is where we get our black and white contrast. If the area of the cell, the receptive field for the ganglion, is in the center, it's going to cause excitation of the cell, more action potentials, more information to the brain. If it hits the surround, you're going to decrease and you're going to get dark. So think of it as black and white. In the center, you're going to get white. If it hits the surround, it's going to inhibit and you're going to get black, the opposite effect. This is similar to how the color ganglion cell receptive fields are. Only instead of black and white, it's red, green, yellow, blue, etc., etc. So you may be looking at this slide, and on the left-hand side, you see about 16 boxes with a lot of white in between them, separating those squares or boxes. And if you feel your visual system is playing a trick on you, it's no trick, I assure you. In between the areas of black and white, and I'm sure you've heard this analogy a lot, is gray areas. So what your brain is doing is it's trying to figure out the contrast between the white areas in between the squares and the black squares themselves. And so what it appears is that your visual system's a little confused. So what it's seeing on screen is little gray dots in the corners in between the black and white. And what this does is it shows us this idea of what we call lateral inhibition. So looking at the chart on the right, on the left-hand side, which is the y-axis, we see light intensity going from low to high. And we see distance on the bottom. Now, looking at what we call Mach band, so this is something that's called the Mach band illusion, on top of the graph, you see 4 shades of gray, basically, in B. The lightest shade on the left is sort of a light gray, and then a little bit darker as you get to A. B and C is the border between the second and third shade of gray. And then D, after D on the right, is the darkest shade, like a charcoal gray. What we are trying to attempt to explain, or what I'm trying to attempt, and the book is trying to help me with the slides, is between B and C, we can see that edge where B is a little bit lighter and C is a little bit darker. And something weird may happen if you're really concentrating. A, which is in the center of that second shade, seems to be a little bit different between the border of B and C, and D seems to be a little different than C. So A and B together are the same color. They're in that same shade. C and D together are the same shade. But there is an interesting phenomenon that happens if you really concentrate in on the line between B and C. When you're looking at them next to one another, B is lighter compared to the darker border of C, and so B appears even lighter than A. C is a little bit darker than B, and so C actually appears a little bit darker in our perception than D, even though A and B are the same shade, and C and D are the same shade. So in terms of actual light, if we look at the first graph, A and B are the same shade, so a little higher light intensity. C and D are a little bit darker, so a little bit lower light intensity. But our visual system, and this idea that we call lateral inhibition, shows that a light line next to a dark one makes the light one look lighter, and a dark line next to a light one makes the dark one look darker. Hence, even though A and B are the same shade, B looks a little bit lighter because it's compared next to the darker shade of C. And C looks a little bit darker than D because it is right next to and compared to the line of B. So this is something that's called the Mach band illusion. So even though A and B are the same shade, B compared to C looks a little bit lighter. C compared to B looks a little bit darker. So our perception is that B is a little bit darker than A, and that, B is a little bit lighter than A, and C is a little bit darker than D. And that is part of this thing we call lateral inhibition. Obviously, we don't see lateral inhibition. This would have been a really good slide to show that, but it is not there. So I will do you guys a solid and go back to this area here. So this is what we're talking about right here. Looking at the receptors that we have, here we have the stimulus. We have a lighter shade of gray and we have a darker shade of gray. And then right here between 7 and 8, that's the line between the two shades. So what we see is that there are 15 receptors. We can call those rod receptors, and then they are going and giving their information to the ganglion cells here. So when that information sharing is taking place, let's say that number 7 is going from the rod to the number 7 ganglion cell. Lateral inhibition means that if 7 is hit, it inhibits 8 and 6. If 12 is hit, it inhibits 11 and 13. So basically, the cells that are excited are inhibiting their neighbors. And because here between 7 and 8, there is a dark light contrast, what we perceive is that the lighter area right next to the darker one looks a little bit lighter. The darker area next to the lighter one looks a little bit darker. So even though there's no difference in the stimulus, the way that our brain perceives it makes it look like the light one is lighter next to the dark edge, and the dark edge is darker next to the lighter edge. This allows us to understand edge detection, where one thing ends and another thing begins, and how we can distinguish between different shapes, different objects, and things like that. So here you can see how does lateral inhibition work. That video is going to be a link in that accompanies this lecture. So let's head back to the main lecture here. Thank you for hanging with me. All right, so now we see the effect of light on center and the surround of the receptive fields. Again, we are missing the picture, so let's go back. That's why I have this. And here we can see what it looks like in a smaller slide. For the visual fields, the receptive fields of the visual system for the ganglion cells, we have the one that I talked about earlier. So if I go back up, we saw this right here. We saw the on-center, off-surround, right? So if light hits the center, it excites the cell and it fires more and you get more light. If it hits the surround, it inhibits the cell and you get more dark. And that gives us our contrast. Lateral inhibition just says for the cell that fires, its neighbors are going to be inhibited, right? So we'll be able to see that difference and be able to sharply detect the two different shades. Looking here, we see that there are two different types of ganglion cells. There are light in the center increasing the fire, just like the ones we saw, called the on-center cells, where if it hits the surround, it reduces the firing. But we also have the opposite. We also have off-center on-surrounds. So we have both types. In the off-center cell, light in the outside, right, will cause it to fire, but hitting in the center will cause it to inhibit. So there's also another one here you see called Mach bounds in our perception of values. I will make sure to provide that to you as well. So we have on-center, off-surround types of ganglion cells, and we have off-center, on-surround. Let's go back and hope that we have more slides that actually work here, that actually show the picture. So when we get information that goes through that primary visual pathway, information that goes through the LGN to the visual cortex, or V1, primary visual cortex, we actually don't see the big picture yet. Just like in the auditory system, it has to go to secondary association areas for us to put all that information together. The information that comes just into V1 is only giving us information about line orientations from a cell in V1 called the simple cell. So when information first comes to V1, it's very elementary. All we're looking at is understanding the orientation of different lines, horizontal lines versus vertical lines. So there are separate cells in V1 that correspond to horizontal lines and will fire, and there are ones that respond to vertical lines that will fire, and there's ones that, you know, perpendicular. If it's not what the towards the orientation of the cell, you get no firing. So you see no little lines that are representing action potentials. If the line is diagonal, as you can see in the next one down, there's a little bit of it that orients, but not a lot, that orients vertically. So you'll get a couple of little action potentials, pop, pop. When it's actually lined up the way it's supposed to be, that's where you'll get maximal firing. So what you see there is brr, pop, 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 right? That's that cell giving off action potentials because it is detecting an orientation in what it's designed for, vertical. If we look at horizontal the other way, we get, again, just a couple that go through that vertical area, and so we'll get just a couple of little responses, a couple of little action potentials, bop, bop. Now, it doesn't matter if it's a vertical line that's perfectly centered, like in the middle, because if you look at the bottom, we have one that's also vertical, but it's not centered in that little target. It doesn't matter if it's centered or where it is in the retinal field. As long as it's a vertical line, you'll get pretty much the same response from that cell, right? So that's simple cells in V1. They only respond to a particular line at different orientations, vertical, horizontal, and we get some sort of input for diagonal and circular and things like that. So the combination of different simple cells gives us sort of our idea for edge detection or the shape or outline of certain things. Again, this is, by the way, from a cat's striate cortex. So it's from the visual field of a cat that was used for this study. But that's why, again, in the dark, sneaking home after midnight, you're able to see the outline of things like the couch and the coffee table so that hopefully you can avoid hitting your shins and making a loud noise and waking everyone up. So here's another one that we can't see. Simple cells of the visual cortex. So again, ganglion cells get information from the rods and the cones, and they overlap with the receptive fields. So this allows them to respond to a line or edge at a specific orientation. So we talked about what the simple cells look like in the cat retina. Now let's go down and take a look right here. So here, and thank goodness for this, or we would have had a tough time with this lecture. You can see that the simple cells, right, have input. So ganglion cells overlap with the receptive fields. So here's the receptive fields in the retina, which send their information to the ganglion cells, and that information from the ganglion cells combines for these signal cells. So as I wrote here, when information gets to the visual cortex, or V1, from the ganglion cells, the only information processed at this level is about the way the lines are oriented. The cells in V1 are simple cells, and they carry only information about line angles. They're the ones that respond to their preferred orientation. The vertical ones from the last slide, right up here, will fire the most. The ones that are, the ones that don't, are at a different orientation. And it's the combination of these simple cells that give us the outline of things in the real world. So the vertical hatch marks represent neural responses. The yellow line underneath indicates when the stimulus occurred. And notice that the response is greatest when the line is closest to the preferred orientation, and it's least when the orientation is most discrepant or away from. In the last example, the response was diminished because the stimulus did not cover the cell's field. So, looking at simple cells about different line orientations combine in V1 to give their information to a little higher up type of cell called complex cells. So complex cells take the line orientations of the different simple cells together and try to create a bigger picture. So, as it says here, simple cells get their information from the ganglion cells in that retinotopic fashion. From V1, simple cells give all the information about line orientation to the complex cells. The complex cells receives information from the several simple cells, and each serves to group in sort of a circular field fashion. As a result, complex cells tend to respond to things like different types of illumination, and as the edge moves to the left or right. This also helps us to see something moving in a still background, which means this is what helps us to detect movement. These cells get a little more information about the visual image, which accounts for detection of boundaries or lines that separate one thing from another. We still don't know specifically if we process information about texture in these cells or when simple cells give their information to complex cells. Outside of V1, complex cells give that information then to hypercomplex cells. So what you're seeing is the sort of hierarchy in this case of higher order processing. So we see form, an outline, which gives its way to different specific shapes, and that gives us more information that tells us what something is. We see light and dark contrast and things like that. So let's see what we got here, if we can get to the next slide. The next slide, again, we don't see much here, so I apologize for that, but we talked about in the auditory system this idea of a Fourier analysis, where basically what the cells are doing is taking information, and remember we talked about the difference between sensation and perception. In sensation, we called it a bottom-up process, where you're basically getting individual puzzle pieces. So think about these cells that we just talked about. Simple cells, complex cells, ganglion cells, all of them are breaking up the information into different orientations, different shades, different colors, and basically breaking apart the scene and putting it back together. So color is really added sort of later on. But this idea of a spatial frequency analysis or Fourier analysis for luminosity or light is the same as what we would say for chapter 9 and the auditory system, where it took separate things and then put them together so that you could hear a band together when it's perceived, but you could also pick out specific instruments like the clarinet with the overtones or the drums or a guitar, right? In this case, you're taking lower and higher spatial frequencies, and it helps you to separate the big picture from like these separate lines and things like that. So we can detect edges and gradations of change by taking the information apart, and simple cells and complex cells and different types are all basically interpreting it separately and then putting it together in the big picture for what we perceive. So looking at this, you can see that high frequency transitions may not be very meaningful if you don't have low frequency transmissions, and maybe vice versa. So here's the original picture. You see a little kid with a little white teddy bear. And if we remove the low frequencies, which is sort of the, remember, blurry outline of things where you can kind of tell what's in the foreground versus the background, or when one shape ends and the other begins, if you remove all the low frequencies, all we have is sort of a black and white silhouette. You can really see detail in the lines of the child's hair, the teddy bear, the child's eyes and ear are very easily seen. And that's with the higher frequencies. So again, those would be more of the hypercomplex cells that take information in. If we look at the one on the right with the high frequencies removed, we see a pretty blurry picture, but it's still pretty easy for us to tell the child and the child's bear or little toy from the background, which is a lot darker. We can see the outline of the shirt, we can see the skin, we can see the hair as it's kind of frizzy, and we can definitely make out the white bear from the dark background behind it. So the lower frequencies are responding, and you see sort of the blurry shape. The higher frequencies is more of like the silhouette that you see in the middle. And so what our brain does is it takes the information from simple cells, complex cells, hypercomplex cells, and it starts to put everything together. So here would be the rods and processing of black and white. If we have the cones involved, we have the nice, sharp, clear original picture and potentially adding color to it and everything. So that's sort of how it works. In the visual system, very similar, again, to the auditory system, as I promised to point out. So we tend to process things, perception of objects, color, movement. When we process things, we call this parallel processing. My apologies for my overactive inbox of my email. So we process things in three sort of ways, right? We tend to process things in a modular way, which means we segregate brain functions into separate locations. So again, those simple cells and complex cells are in V1. We have V2, V3, V4, V5, and other structures. And so information is there. So line orientation, and outline of form is V1. V4 is where you add color. V5 is really where movement comes in. So it's modularly processed in different sort of sections of the visual cortex, and then that information is put together into the big picture. It's also processed, as we've just talked about, hierarchically. So the first thing that we process is lines. The second thing that our brain processes is putting them together in a shape. Then later on, we can see the contrast, and we can see movement, and we can see color, and it sort of adds all of that. So the most basic things like line orientation are first. Those simple cells are going to go first. And as the information spreads, it gets more hierarchical. It gets more higher order. And the more complex it gets, the more of the big picture that we see. It also means that it's distributed. And this is sort of where we're going to come back to that idea of the two systems, the dorsal and ventral streams. So processing is also distributed. So it occurs across a wide area of the brain. This also allows our five senses to interact with one another. As we saw in the language chapter last time, information for language had to combine visual information for reading and auditory information for processing language. And the visual information for people, especially if they're deaf or that learn sign language, through gesturing. So another view is that, like language, vision is a mix of modular and distributed processing, where different areas do different things. The angular gyrus, right? From there, Wernicke's area, the visual cortex, right? So it's sort of a mix of all of those things. If we think about it, right, when we watch TV or go to movies, it's obviously the visual system and auditory system working at the same time. In the movie theater, you see the movie coming off the screen, and you hear the sound coming from the speakers, and it gets really annoying if they're not in sync with one another. So usually they are, and there's no problem with that, right? So it's a mixture of these things. So that means that we have information specifically coming from rods with a larger receptive field, and we have information that comes from the cones through the ganglion cells to the brain, which is much smaller receptive fields. So that gives us two pathways of visual analysis. That gives us the parvocellular system. Parvo means small, so remember the ones with the small specific direct receptive fields were the cone systems. So that gives us, these ganglion cells are located specifically in the fovea, that area of central focus. They give us small color opponent circular receptive fields, those ones that we talked about in color vision for the opponent process fashion. So we have for red-green, for yellow-blue, for black-white, we have on-center, off-surround, off-center, on-surround, depending on where it hits. For the magnocellular system, that would be the rod system. Magnocellular means big, magno means big, and so we have larger receptive fields on the ganglion for the rods, because remember, it's not a one-to-one process. It's many rods giving information to one ganglion cell. So these are the cells in the periphery. They have very large brightness opponent receptive fields, like the black and white ones we saw at the very beginning of this lecture. They're very fast on and off because we need to detect the difference between light and dark. And that tells us the contrast. That's the Mach band illusion, the lateral inhibition. It gives us the brightness contrast so we can see where one object ends and the other begins. And it can see movement of showing movement across a still background, or if many things are moving, we see the contrast between them. I'm not sure if this one had a picture to it. I don't think it did, but let's see if we can go back here really quick. And come back to me. There we are. Here we go. And just, this is our sort of fail-safe to make sure that we can see if there was anything to that slide. And here we see, nope, nothing there. Good, just making sure. Is there anything that I left out that I might have typed up? As we've established, the rods send information to the ganglion cells, and the rods have a larger visual field. So again, this system is called the magnocellular system. It's in the periphery, fields are large, and we have the brightness opponent process. The information from the magnocellular system, this is what goes up into what we call the dorsal stream. That's the where system. So that's telling us if we can see movement and if we can see shape, that tells us where in our visual field that something is. Remember, we had the dorsal and ventral stream. The dorsal stream in chapter 9 was also the where system that told us where a sound was coming from, which direction. That helps us if we're hearing a siren when we're and we need to decide whether we need to pull over or not, or if somebody's calling you, what direction they're calling you from, especially if they're in danger. So that's the dorsal stream. This magnocellular system goes up towards the parietal lobe to the dorsal stream, whereas the parvocellular or smaller system, parvo means small, smaller visual field, receptive fields, is using those color opponent cells that we talked about in the opponent process theory, and the information from this system about color and recognition of specific stimuli goes to the ventral stream. And remember, the ventral stream is the what stream, so we're about to get to that. And that's the one that stays low through the temporal lobe and goes to the prefrontal cortex. So, going back, here we see the ventral stream, or the what stream. So this one, as you can see, if we were to zoom in, you see V1 at the very back of the occipital lobe in pink, and you see V2 right above it in green, and from there it goes into the areas like, you can see the angular gyrus that it's connecting to, or down below on the bottom of the temporal area. Remember, we have superior and inferior, so V4 is on the ventral surface. We see the inferior temporal cortex, and then that goes up towards the prefrontal cortex. So this ventral stream, which stays low, ventral, towards the stomach, right, bottom of the brain, stays low in the occipital lobe to the temporal lobe, and then goes up to the prefrontal cortex. So V1 through V2 to V4, and then this inferior temporal cortex. This allows us to know what an object is that we're looking at. So we have color perception and object perception. If I were to hand you a red pen, or if I were to hold it out in front of your face and say, what is this? The ventral or what stream allows you to understand that is a pen, and it is a red pen specifically. If you were to have damage to this stream, and that damage does not have to be on the pathway from the eyes to the brain, but it could be in V1, V2, V4, the inferior temporal cortex, then damage in that stream, in that pathway, people would be able to know where the object is, but they probably wouldn't be able to name what it is. So if I were to hand it to Dr. C, he would be able to take it from my hand, but if there was damage to his ventral stream and I said, what is this object? Dr. C wouldn't be able to tell me. He would just say, I think I know what it's for, but I can't name it. So he would be able to grab it from my hand, but he would not be able to know what exactly it was. So I just took a pencil from him, if I may demonstrate, and if your ventral stream was damaged, you'd be able to grab this pencil from me, which he's doing, but if I were to ask you, what is it, you wouldn't know. So what would you say?
I think I know what it's for, but I don't know.
There you go. So next is our dorsal stream, which again goes up into the parietal lobe, goes dorsally towards the top of the brain to the parietal lobe in that posterior parietal area, and then over the top to the prefrontal cortex. So in this case, right, this is the where stream. So in the auditory cortex, we also had the where stream, and it told us where a sound was coming from. In this case, you're looking at where in your visual field, how far away something is, so distance, and being able to understand your body relative to where it is. So that idea of going up to V1 to this area called V5 or MT, which is medial temporal area, which is sort of the angular gyrus too. And it goes, instead of across, it goes up to the posterior parietal lobe. If you recall from earlier lectures, the posterior parietal lobe, the parietal lobe itself from chapter 3, was about paying attention to stuff and knowing where we are in space. So knowing where we are relative to our environment and knowing where we are relative to an object, such as a car coming towards you, or again, handing Dr. C a pencil. So V1 to V5, and then to the posterior parietal lobe, which not only tells us where we are in space, but tells us where this object is relative to where we are. So in this case, if there was damage to the dorsal stream, Dr. C, if I were to ask you, what is this thing?
The pencil.
He's able to tell me that it's a pencil. His ventral stream is fine. He can tell me what it is. But if I were to hand it out in front of him, he would have a lot of trouble trying to grab it. He wouldn't be able to actually know how far away he was or sense how far away he was from the pencil. So he might grab for it and he may miss several times and maybe he'll get lucky and reach out and grab it. But it's really hard to tell where an item is relative to where you are to be able to reach out and grab it if there is damage to the dorsal stream. So the ventral stream is the what stream, knowing what the object is by looking at it, or conversely by hearing it, knowing that it's a voice, and specifically that it's your mom's voice telling you that it's probably lunchtime, Dr. C.
Hi.
And the dorsal stream of knowing where the voice is coming from. Oh, it's coming from the kitchen, so he better go because she's already made it. For the visual system and the pencil example, the ventral stream is knowing what the object is. If there's damage, he'd be able to grab it out of my hand, but wouldn't be able to name it as a pencil or pen. Damage to the dorsal stream, he'd be able to say pretty easily, oh yes, that's a pencil, but he would have trouble grabbing it from my hand. So again, whether it's the ventral stream or the dorsal stream, they both take different pathways through the brain, but they meet up in the same area, the prefrontal cortex, which again is responsible for putting all of the information together from the five senses and being able to understand and compare it to memories and things like that to know what to do with that. That's our perception of it. So it manages information in our memory while it's being used. We're going to come back to this concept of working memory in chapter 12. So for example, it integrates information about the body and objects while planning movements. So Dr. C wants that pencil because he then wants to go and draw something. So what he has to do is plan out his movements. He sees me holding out the pencil. He reaches out and grabs for the pencil. And then he walks over to his desk where there's paper and proceeds to sit down and draw a picture or write something down in a story. And so for him to be able to do that means that he has to know where the object is, the pencil. He has to be able to reach out for it. He has to know how far away he is from his room and make steps towards that. So that's movement. That's the visual system. Hearing, or if he realizes that he can't start his art project because mama wants him to eat lunch, then he has to put down his pencil after hearing mom, and he knows he has to go eat his lunch. So you're integrating information from the visual system, the auditory system, touch system to be able to move your arm, walk to the next room, and plan all of these things out at the same time. So that's the prefrontal cortex, combining information from all five senses. When we're done with this chapter, obviously, we are going to do a shortened version of chapter 11, which is the movement chapter, to finish off this unit. So that will only be, is only designed to be a couple lectures in itself. Looking at disorders of visual perception, if that inferior temporal cortex that you see down below as part of the ventral stream is destroyed or is damaged somehow, cells specific in that inferior temporal cortex, there are specific cells that look at geometric figures so that you know what you're looking at, is a triangle, a circle, a square, a rectangle, and things that are made up of these shapes that we've learned to be put together are things that you can recognize. So you would have trouble looking at faces, hands, words, houses, right? think about a simple elementary school drawing of a house. We see a big square as the main part of the house. For the roof is a is a triangle on top of that. The front door is probably a rectangle in that, and the front window could be broken up into a couple of little squares, right? So what our brain sees first is square, square, rectangle, triangle. But when it's all put together and sort of superimposed, oh, that's a house. A bunch of lines and shapes make a chair. Words are made with different letters with different shapes, right? And things like that. Um, Seeing people's faces in the inferior temporal cortex are things like this. So we do have this idea that we have to learn people's faces. We learn language. We learn words. So the capabilities to recognize the different shapes and things like that seem to be hardwired. We seem to be born with them. But just like we have to use our visual system, our auditory system, and we have to strengthen the synaptic connections of these pathways from the ear to the auditory cortex, from the eye to the visual cortex, and things like that. Our capabilities to do so are hardwired, but we have to learn and strengthen these connections in order to get them, or we just lose them. So, learning faces and things like that. When we get into chapter 12, we'll talk a little bit about Alzheimer's disease and how some of these cells in the frontal lobe and some of these cells in these pathways can be destroyed. So people with Alzheimer's may not be able to recognize faces of people they know or remember things from their past, right? So damage to this ventral stream shows that people may have trouble in recognizing what an object is. Specifically for faces, there's an area called the fusiform area of the, or fusiform face area of the inferior temporal cortex. So if I go back really quick, again, down here at the bottom, I'm going to move my cursor over this area, the inferior temporal cortex. There is an area specifically called the fusiform face area. And what the fusiform face area, here you see sort of in yellow, on the right side is that there are some people that have genetic defects, or if there's brain damage specifically to that specific area of the brain, it causes a disorder that we call prosopagnosia. And prosopagnosia is the inability to recognize faces. So if this genetic defect takes place, or if there is brain damage specifically to that fusiform area, you will not be able to recognize faces. So Dr. C would come home from school, or in this case, since it's homeschool, come from the other room to my room, and I would say, "Hey, what's up?" And he would look at me and just not be able to recognize me. He wouldn't know who I was. However, of course, this is only for visual recognition. So if he were to close his eyes or maybe just pay attention to my voice, he would be like, "Oh, that's Dada. That's Dr. B," because he'd be able to make the connection from the auditory system. So there was a study called the Griebel study, which showed that this area of the brain is responsible for non-human and human face recognition. So it's not just recognizing human faces, but recognizing your dog, recognizing other animals and other faces. So here is this Griebel study, which showed. So here's an example of Griebels that kind of look like little statues. They're kind of look like faces where the it looks like maybe a tongue is sticking out or something like that. They kind of look like gargoyles, right? So looking at these fMRI scans, you can see that the place where faces are recognized light up really bright red in people that have, so they have Griebel novice and Griebel experts, right? So looking at the top left, Griebel faces, we see a lot of lighting up because this area of the cortex recognizes faces. Looking at Griebels, when you've never seen them before, they don't look like faces, right, necessarily, until you really get used to them. So you don't see those same parts of the brain in the fusiform area light up where activity is going on. So they're not really recognized as faces. When for people who have had a lot of time with greebles and they can make the connection that greebles kind of look like kind of human-like faces, maybe you can see on the bottom some sort of beak, or maybe it looks like a tongue sticking out. Looking at the experts, you don't see much difference between an actual face and the greeble, because you're used to seeing this object and you've made the connection that it kind of looks like a face. Now, if you allow me to indulge, I want to see if I've written anything profound to my class. And looking at the greeble study, I just want to be able to see if I wrote anything. So here in the ventral stream, the inferior temporal cortex, and it contains cells that are specific to detect things like faces, figures, shapes. I don't think I went over the specific greeble study. This won't be on the exam, so I just wanted to see if I had mentioned it in here, and apparently I did not. So let's move on. So I did also mention that in your visual system, unlike the auditory system, which only has really one pathway, and that pathway can be very complex. I kind of tried to limit how much exposure you got with certain things. But for the visual pathway, it was pretty simple. We talked about the primary visual pathway being from the ganglion cells that the axons form the optic nerve. We only really have two stops in this primary pathway, the LGN of the thalamus and V1 that goes to the visual cortex. So that is the primary, what we would call the conscious visual pathway. But I did say that there was a secondary pathway that did go through the midbrain, whereas the auditory system went through the inferior colliculus. The visual system has a separate secondary pathway that goes through the midbrain of the superior colliculus. Remember those little ball shapes that were on top of the inferior colliculus that made the tectum. And then it goes through the basal ganglia, which we learned about in chapter 3 for sort of smooth movement. And some of our eye muscles going side to side and being able to see things is part of that secondary pathway. So it is very possible then, since we're talking about damage, that there can be no damage to the primary pathway, which goes through the LGN and V1, but there may be damage to the secondary pathway that goes through the superior colliculus and the basal ganglia. If that happens, there may be problems with visually responding to written words, and something that we call blind sight. So we know that damage to V1 causes cortical blindness, but damage to that pathway that includes the superior colliculus and the striate cortex, or the part of the basal ganglia, a person can react unconsciously to stimuli without seeing them. So if we think about pathway to the conscious one, leaving the unconscious one intact, what will happen is something that we call blind sight. And in blind sight, I will obviously include a video for this, but in blind sight, a person can have no visual conscious vision, but they can have an unconscious vision. So what does that mean? There was a famous experiment that was done on blind sight where a researcher had cluttered a hallway with things like a tripod and a box top and a chair, and put things in somebody's way in a hallway. But they told the person that the hallway was empty. So this person has damage to the conscious tract, but not the unconscious. So the superior colliculus to the basal ganglia or striate cortex, right? So bear with me here. The person is told, how many fingers am I holding up? And of course, they are consciously blind. So they would say, I have no idea. Now the researcher says, I want you to walk down this hallway. It's an empty hallway, but I just want you to walk down the hallway. So of course the researcher is lying to this person, but not to try to deceive them. to try to see if this idea of blind sight is actually true. So the person is told that the hallway is completely empty, and as you will see in the video when you play it, there are certain objects that make it not empty. And when the person walks down the hallway, not being led by the researcher or anything, in fact, the researcher is kind of behind the patient or the subject, and the person can walk down the hall not knowing that there's objects there, but the unconscious part of the visual system allows them to walk around the objects. So walk around the chairs and the box tops and the tripods and trash cans and whatever's in the way, even though the person can't consciously see, the unconscious visual system is still intact. And so the person will be able to avoid the objects and not even know that they were there. Pretty amazing stuff. If we were to damage V4, which I told you is where sort of color is processed, we're not going to have color blindness in the same way that we have it in the connection between the cones and the ganglion cells. So color agnosia is not the same as color blindness. In color blindness, we may lack some of the red, green, or blue cones, or it may be a problem with the way that the ganglion cells are functioning with the cones. But in this case, color agnosia is the complete loss of the ability to perceive colors at all. And that would be where color processing is V4 of the visual cortex in the occipital lobe. So V4, if there's damage to V4, we're not going to have color blindness where red, green, or blue, yellow is messed up. You won't be able to see colors at all. And here on the bottom, you can see that there is an interesting idea, which in the visual illusion slides that I'll give you will show you that some colors in a red background, this is the same sort of color-coded Rubik's Cube looking thing. In a red background with a red tint, it looks blue, and in a green-blue or greenish tint, it looks more green. Whereas it's a darker red or more pronounced red in a red background. In the green background, you still see red, but it's a different shade. And this is showing us this idea that we call color constancy. So not only do we have different codes and different wavelengths and shape, things like that for color, but the one thing that our perceptual system does, and our brain tries to fill in the blanks when we don't have all the information, is that we have perceptual constancy. So colors in lighter or darker areas tend to be the same. Shapes tend to be the same. If we didn't have this, when a bird flapped its wings and went from one side of our visual field to the other, we might think that it was a completely different thing. But our brain is able to pick out the shapes, pick out the colors, and things like that, and keep it pretty consistent. Otherwise, we might freak out when we went from dark to light and something looked completely different. Or if you saw me giving a lecture and I was standing in front of you, and then I turned sideways 90 degrees, you might think I was something completely different or someone completely different, and that would freak you the hell out. So good thing that our brain provides this constancy. So color constancy, shape constancy, size constancy, all of those things. As things get closer or farther away in your retinas, we still know that they're the same object. Almost done with this lecture. I'm just hoping that the slide changes for me. So movement agnosia is the same thing, but the problem is in area V5 or MT. This is the inability to perceive movement. So that part of the dorsal stream, right, not the ventral stream in this case, the part of the dorsal stream that allows you to follow things with your visual field, especially with your eyes, like think about a test where you have the finger test, if you've ever potentially had a concussion or hit your head, where the neurologist would have you follow your fingers, follow his finger or her finger with your eyes without moving your head. If the area MT or V5 is damaged in this dorsal stream specifically, then you will have trouble seeing movement. Things that move from left to right in the visual field or move around you'll basically just see it as a really bad strobe. So it's like you blinked your eyes really quickly and something went from the left side to the middle to the right side of your visual field. So there was a patient specifically named LM. Again, we strip away any identifying information and just use initials or numbers. So LM had difficulty with that guiding eye and finger movement because it would just look like he blinked and it was in a different spot. So it's really hard for people with movement agnosia to see if somebody or something is actually in motion. It's also hard for them with depth perception to tell if objects are moving toward them or away from them, because it's really hard to tell with damage to that specific area of the brain. And again, we call that movement agnosia. The last thing in the dorsal stream, the last area, is called the posterior parietal lobe. That's actually in the parietal lobe. And we talked about this idea called neglect. I think it was in chapter 3. With neglect, usually we see damage in patients in the right side, so the right posterior parietal lobe, although it could potentially happen in the left side. If it happens in the right side, remember that majority of information from the right brain, it goes to the left eye. So it seems like the left visual field is the one that is damaged. So in this, a patient, and because this is an attention deficit, not ADD, but it is interferes with your ability to pay attention, it's like your brain just sort of skips the other side of your visual field and you don't even know it, which is called neglect. It's almost like your brain just forgot to interpret that side of the world. So here you see a person that has neglect, or we call it hemi-neglect because it's only on one side, half of your visual field, right? So you see on the left that there is a clock, there is a house, and a flower. And the person with posterior parietal lobe damage in a famous experiment was asked to just draw the picture on the left and copy it on the right. So you see the full circle of the clock, right? And you see both petals or both leaves of the flower on the bottom side, but there is a lot of information missing. You still see that the arrows point to an hour and a minute, but you only see numbers basically on the right half other than the number 7. So you see 1, 2, 3, 4, 5, 6, 7, but you don't see the rest of the numbers that add up to 12. For the house, you see the door, the pathway, you see the windows on the right side, and you see most of the full roof, but the whole left side of the house is just missing. And these people are not even aware that they're missing it. They think that they're seeing and they're copying the entire picture. Once again, with the flower, you only see the petals of the flower on the right side. You don't see anything on the left. And this is what we call neglect or hemi-neglect. So this area of the posterior parietal cortex allows us to locate and attend, so put attention on our objects in space. It also orients our body and our environment. So again, it's knowing where we are or how far away or close we are to different objects. So those people with neglect their brain will just simply ignore the other side of the world. Although they report normal vision and they don't see any deficits in what they do, clearly when they're asked to draw it, they simply just sort of ignore everything on the left side of space. So those are the disorders of visual perception: color agnosia, prosopagnosia, movement agnosia, and for the dorsal stream, neglect. This concludes, I believe, our presentation. It concludes chapter 10. And so hopefully I explained it well enough, and hopefully you got what you needed from that. When I come back next week or so, I will go over a shortened version of chapter 11, which is the movement touch system and some of our disorders with movement and touch, such as Parkinson's disease and Huntington's disease. And as always, I will talk to you soon.